be here with you all today to discuss something that is really near and dear to my heart. I'm Dr. Peter Corridon, I'm at Khalifa University, and today we're going to be discussing ways in which we are looking to try to build body parts. I'll be discussing some cutting edge approaches that we're trying, and please remember that these things are works in progress. Nevertheless, please follow me today. So, today we will look at a few things. We'll talk about a growing issue in the UAE, which is really driving this topic. We'll look at some of its effects, and we'll also look at some ways that we can try to develop potential possible solutions in the long term. Now, I'm pretty sure that everyone is familiar with the term diabetes. Well, in fact, diabetes is on the rise in this country. Unfortunately, roughly 30% of the adult population can be considered as diabetic, and far more can be also considered as pre-diabetic. So as you can see that this is a growing problem in this country, in this region, and as you can imagine, globally. Now, scientists anticipate these things to continue, and as these things continue, oddly enough, we are becoming, how can I say, entrenched with other issues that come about because of diabetes. One particular issue relates to kidney disease. And as you can see here, we dive into the kidney, we go inside, we look at the macroscopic, microscopic chunks, and you can get to see the blood flowing through this innate filter, which we call the glomerulus. And as the blood passes by, it goes through tiny, tiny, tiny portions which intercalate with each other and prevent the blood from flowing out, but also support molecules that we need to actually expel, which in turn move by and form our urine. So as you can see, coming back up to this microscopic view of the filter, we expel and eventually form the urine, which is housed in the bladder, and actually in turn becomes expelled when it's time to urinate. Now, all of these processes and much, much more get hindered by the downstream effects of diabetes. People can actually develop something called diabetic nephropic kidney disease, which in turn can be a leading cause of kidney failure, which would be called scientifically end-stage renal failure, which is the complete destruction of normal filtration products and far more other processes that the kidney is involved in. When these things come about, I'm sure you can become familiar with the following: the need for replacement technologies, things like dialysis, which in turn help us filter the toxins that I talked about that come about in our body and help make this blood a bit more useful for us as it continues to perfuse our tissues. Furthermore, when things go awry altogether, the best option is transplantation. Transplantation relates to the idea of finding a kidney from a potential donor, which can in turn be inserted into the recipient. Now, this is all well and good, but as you can see with this graph, life is not that simple. There's a high demand because there's this growing problem that we talked about, but there's a much lower supply. So for instance, people like myself who've been listed in the United States as organ donors would happily give our organs, but the trick is that I have to maintain these organs to ensure that they become useful to someone in years to come. Moreover, as things get worse, this supply versus this demand is going to shift. And I can talk about another thing that is actually causing such a shift. Our good friend here, COVID-19. I'm sure many of us have been inundated by this molecule or this viral particle. Now, interestingly enough, this has been seen to actually further tip the scale in terms of generating a greater shortage, because now COVID-19 has been linked with the development of kidney diseases, which can in turn further support the debilitating effects of the conditions I mentioned before. So I just presented that to give you a greater perspective of the issues that we are facing. Moreover, I'm sure you've seen things like this on TV before. You've probably seen individuals on different types of movies waking up in a bathtub, having a bunch of tape around their stomach in a pool of ice, recognizing that they've actually been cut open and sewn back apart because they've lost their kidneys. These things seem hilarious or far-fetched, but in reality, they do exist because there is a high demand for organs and there is a honest-to-God trade of human organs. So we would like to inhibit this process. Now, inhibiting this process and helping people get the organs that they need, well, what do you think we can do? Many years ago, I trained under an individual, Dr. Atala, and he had this vision of developing warehouses filled with body parts, replacement body parts. So as you can imagine, this was really far-fetched back then. But think about it. How cool would it be? How wonderful would it be if we can actually now mimic what we have in terms of car parts with body parts? So in terms of strategies for us to get to that point, we have to start with the following. We can look at human cadavers, we can look at large-scale animals, and in so doing, we can harvest these organs and take these organs, and now we can't put these organs back in directly in many instances because there's something called bioincompatibility. Even if someone gets a kidney transplant, let's say from a twin, there's still the potential for this kidney to fail. Now, how interesting would it be if we take this concept? We take the harvested kidney from a cadaver or even possibly a large-scale animal which has a kidney of similar size, function, and structure. Take out all of the inherent cellular material that can potentially drive incompatibility. We do this by a process called decellularization. That is correct. D, moving away or removing cellular components within this organ structure. And we end up with something called a scaffold, which you see here as the finished product. Now, this scaffold basically consists of something terminologically I can define as the ECM, the extracellular matrix. I can just put this as basically in terms of a cushioning and also support mechanisms. But it's also housed with a bunch of different biofactors or important chemical molecules that drive and support normal function. And with it, it surrounds this thing called the vasculature, or the blood vessel highway that I like to say. Now, we have this, and I kind of liken this to an apartment complex. Each and every time someone moves out of an apartment, they take out all of their furniture, they strip the apartment down, and they're left with just the basic structure. The floor, the ceiling, the surrounding walls. You can liken that as a scaffold. And all that space would be the vascular highway. The structures that I mentioned, the floor, the ceiling, the walls, you can consider that as the extracellular matrix or the ECM. When a new person has to come in, we have to repopulate that. So thinking of this concept and applying this now to the building or the re-engineering of organs allows us to come to this. So many years ago, while I was thinking of the whole concept of regenerative medicine, building body parts, repairing body parts, we first started off with the idea of repairing body parts. And ways in which we looked to do this was inserting different genes, trying to repair things from the microscopic perspective. So as you can see here, we have a bunch of different factors, important chemicals that can be introduced into this kidney. And you can introduce it into the vascular highway. You can put it into a large vein or a large artery, or even into the ureter, which is at the bottom of this figure. The ureter is the end point where the urine actually starts dripping out of the kidney and forms. So you can think of this, and as we thought about this, we decided, why would it not be possible to take the scaffold, introduce a bunch of these compounds and factors, and try to rebuild this? Rebuild this in such a way that we can take a potential patient's own cells and build up a kidney that may be customizable or customized for said individual. So that was our hope. That is today still my hope as I push forward with this research. And my main interest related to the following. I wanted to take the concept of looking at the intrinsic structure, intrinsic function, as you can see with the human model, go into the laboratory, and in so doing, introduce this scaffold, introduce this potential bioartificial kidney that we're trying to build. And as we push this in, we wanted to see how it would function inside of an animal. So we transplanted this. And oddly enough, we gained a lot of information related to its basic characteristics. So characteristics relate to its failure rate and its inability to support innate structure. and also innate function. Strictly speaking, the actual scaffold popped under the high pressure of the blood within the system. Also, it provided a bunch of incompatibility restraining factors or responses from the animal. We expected this to happen, but it was great to actually be able to define and characterize this. The next step as we re-engineer or reverse the process was to take this and develop something we call an in vitro model or a simplified model. In this you see here, I have this large pump to the left. And with this large pump, I actually simulate this as the heart. And as you can see, a bunch of tubes going in. And this relates to the thorax. So I take my kidneys and I run them through with a bunch of different chemicals. I started off with saline. And then from saline, we went off with blood. So basically you can see here, we are taking this step by step by step to reintroduce the different compounds, reintroduce the different flow rates, and characterize things as we go by. Interestingly enough as well, we developed the failure rate for this system, which is great because these are intrinsic points that we need. We developed mathematical models to basically understand how the inflow and the outflow relate. And in so doing, these things are going to help us once more drive this function. So I just played this video again for you to get to really see and understand and really, how can I say, appreciate what we've done in that case. Moreover, a means of trying to see and investigate this, we started looking at things called angiograms, or basically the vascular structure and how it changed under these conditions. So this would be the normal vascular structure, or we call this the vascular tree of the entire organ. Interestingly enough, once it was put under these conditions, the entire vascular tree was disrupted, which we expected. But we were able to define this as a function of time and as a function of rate. So we were able to not just look at things within the animal, not just within the in vitro model, but characterize both cases in terms of failures. And so doing, we were able to start looking at things from the microscopic level. And looking at things from the microscopic level, as you can imagine, you would have to go on to a smaller type of animal system. So we moved away from the porcine system and we went into the rodent system. So looking here at our good friend the rat, we inserted these things and you can start looking and seeing failure rates. And more interestingly, you can look at a really high-powered technique which allows you to see inside the actual scaffold or inside a kidney in real time. And you can color code this and you can get to see how fast the blood is flowing and you can get to see how it's vibrating with the blood pressures. Interestingly, you can also see remnant cellular material that was also reintroduced into the system. So these are some high-powered techniques that can potentially help us as we move forward with this technique. Now, in summary, I just want to remind you that I introduced a topic that related to diabetes. We defined the issue that we're having. Moreover, we looked at the downstream effect, kidney disease or end-stage renal failure. And lastly, we looked at something that we're really trying to do. We're trying to hopefully create an organ by re-engineering an existing organ. And in summary, what I would like to do is give us a fighting chance, not just here in the UAE, but potentially globally, as we try to fight diabetes and develop far better technologies to support transplantation. I thank you so very much for allowing me to spend these few moments with you, giving you an idea of the interest that we have here at Khalifa University, as we have here in the College of Medicine. And in so doing, I would like to give you all an opportunity now to ask whatever questions or even provide me with comments as we go forward. Once more, again, I am so grateful for this opportunity and I thank you. So allow me to read some of the points or questions that I'm getting, and I would hope to try to engage you all as this process continues. So I have one question here that comes from the audience. How is it possible to do kidney transplants from animals to humans? So first of all, there is this idea of xenotransplantation. So now, as you can imagine, it is possible because again, you have the idea that the kidney is the same size, it's the same function, but you also have the inability or issues that you might get related to compatibility. But if you strip down the structure and you reintroduce cells from a specific patient, the aim is to hopefully suppress that immunocomprehension or immunocompatibility or issues that can arise from different species by enhancing the own person's effects or the own person's cells and the own person's compatibility. So that's one way. I have another question I can look at. Wouldn't a kidney fail after a few years due to the immune response? That's a great question as well. So we do not know. So what we're trying to do as well is put in, again, this is a long-term process, put in ways in which we can actually have this system be a bit more aligned and in so doing, potentially work better with the body that it gets transplanted. Now I have another point here. Thank you for the interesting presentation. Well, I thank you for this comment. And the question is, do you see a brighter future for diabetes research? Well, I think there's always hope. And if there are not people like myself, people not like yourself, interested in doing these things, then there is no hope. Rest assured, these things are being addressed by numbers of scientists all across the world. So this is something that we're really trying our best to, and we're trying to develop novel, really interesting ways in addressing these problems. So again, I have a lot of hope, and perhaps one day we can get ahead of these diseases. I have a question here as well. I have IF/TA is a major problem in kidney transplantation. Can you replicate such a fibrotic situation in your experimental setting? I'm not really sure what IF or TA stands for, so forgive me if the person can, yes, if they can outline that, but I can at least address the fibrosis. So in terms of fibrosis, this has to be replicated once we actually enhance the cellular structures that come in. So yes, this would be possible as we build up the model. So initially we wanted to just look at the failure rates or the destruction that comes about with the scaffold itself. Now we've also introduced different things related to different types of cells. We started looking basically at the endothelial or the blood vessels, or blood cells if you will, to form the basic structures with these. Now as you can imagine, you would have things like clustering and sloughing off of different types of cells, which in turn can actually cause occlusions and mimic effects as you can see with fibrosis. So yes, that answer is possible and we are continuing to work on that. Now, what is the view regarding 3D printing? Well, in terms of 3D printing versus what I've discussed, 3D printing is an excellent option. Interesting thing though with 3D printing, when you start looking at 3D printing, you have to start from complete scratch, so from the bare basics. And as you build this up, like this technology, one thing that actually inhibits both systems is the vascular highway or the vascular systems that can be incorporated into them. It is possible to do this with 3D printing. Now, one benefit that this has over it is that this decellularized scaffold contains all the biofactors within the extracellular matrix. You can't have that in the 3D printing unless you actually embed these things in it, which in turn increases the complexity. So again, I'm not saying that it's not possible, just understand the varying degrees as we go by. And I hope this answers that question. As I continue, so are there artificial kidneys such as the artificial heart? Excuse me, sorry. So at present, these are all models that are being developed. So when you say the artificial heart, so they've been looking at, how can I say, there's the bioartificial and then the artificial. So they've been looking more at the mechanical systems which can actually be mimicking these things. And there's a group out in San Francisco that have actually put forward something that I know is being fast-tracked as an artificial kidney. So yes, these technologies do exist and they're coming of age. Are there side effects of kidney transplant for the donor to the long term? Absolutely. So you have bioincompatibility. You also have something that we've generated in our model. You have something called stenosis. Basically, the artery, the renal artery getting clogged and so forth, basically starving the actual organ of blood and thus nutrients, and being unable to in turn take away waste products. So that's one long-term effect. And these are things that we're trying to overcome. Not very easy, but again, these are issues that scientists are aware of, and we're trying to figure these things out. I have one question. Have you tried co-culturing decellularized scaffolds with human cells? Will the organ work as expected? So we're not working with human cells at this point. So the main aim is to actually determine what can happen within one specific species. So the aim would be, first of all, if you can get a scaffold, if you can get a kidney, for instance, take it from a given animal, strip it down, repopulate it with the animal's own cells, and then see how that is going. Then you can start looking at crossing with different types of cell species, etc., etc. So I think the first stage for us is really focusing on same species and looking at the issues that relate to that. But this is where we will have to go in the long term. Thank you. You're welcome. I'm looking at one other question here saying, can you once again please elaborate what was the effect of the entire process on the vascular tree? Absolutely. So what I will do is, if you don't mind, I can go back a bit. So would it be all right for me to go back? I want to show you this. So take a look at this. So this is initially really great from what we saw. So we have now saline perfusing this organ. And as you can see here, you're kind of getting this pulsating effect at the renal vein and the outflow at the renal, pulsating at the renal artery and outflow at the renal vein. So as it's twisted now, you can get to see the ureter or the outflow for the urine at the end. So this was great initially, and this is what happened when we left the kidney at the beginning of the experiment, and we let this traverse for about 24 hours. And in terms of the effect that we saw, well, after a while, you saw the vascular tree being completely disrupted because you actually had a degradation or breakdown of this extracellular matrix. So think about it. You have a gush or a massive amount of flow, high pressures, and the systems there, they are not designed so far to take that on their own. But for us, that was really interesting because that gave us an idea of the failure rate and what we need to overcome. Because again, as my mentor many years ago said, the aim for regenerative medicine should not be to try to rebuild the exact same system. Would it not be even better to try to develop or generate something that may be a bit more resilient to these types of diseases? So that's what we're looking at, and I hope that answers that question. Allow me to continue. Does the technique work for the kidney only, or can it be applied to different organs? Well, when I trained in my postdoctoral fellowship, the good thing was that we were in an entire 6 to 7 story building that was designed and catered to building every body part in the human. So to me, every day going to work was just, wow, was just enlightening. Because I know that I could have taken my love for the kidney, my interest, and then lower down the hall, I can walk down and talk to one of my friends as they explored their love and their ability to try to regenerate the lung, the heart, the spleen, etc. And actually one of these compounds that we actually devised was in turn developed and has been transplanted into a living donor. And interestingly enough, once I was leaving my postdoc, that individual actually came into our laboratory and he started training under us. So hopefully we had a nice little loop and the circle continues. I have one other comment. Thank you for the interesting field of research. I appreciate this. Thank you so very much. I have one other point here asking, will the organ work as expected? Well, that's a really interesting one. And I can say that I really am not sure. Because as you can imagine, this is one of the interesting points that I looked at. I think the kidney is one of the more complex organs in terms of its structures and functions. So I think once or if we get a chance to rebuild this in any appreciable way, these things can be applied to different organs and hopefully we can develop technologies that can address and help us better build bioartificial organs. Okay, well, I would like to again thank you all for all these wonderful comments and also these exciting questions. And my information is available. I would be happy if anyone would like to contact me thereafter. I'm always interested to talk to someone who has great ideas, interesting ideas, and also just interested in finding out more as we move towards, hopefully, this new age where we can address different issues within the body. Thank you so much again. I'm Peter Corridon. Have a good day.